All right. So thank you for missing some of this gorgeous weather and coming in to listen to this talk. Thank you for coming out here. My name is Derek Morelli. I'm the chemistry instructor here at SWOCC. And I have a brief introduction, but first I want to let you know about our next talk as well. So on April 25th, we will have Dr. Alexander Ruzicka coming to give a talk on expecting the unexpected, astro serendipity in the Cascadia Meteorite Lab. So that will be on April 25th. But for tonight, we have Dr. Cory Brooks coming up from Fresno State, or California State University, Fresno. He's a faculty member at or in the chemistry and biochemistry department. He also serves as editor-in-chief of the Journal of Monoclonal Antibodies and Immunodiagnosis and Immunotherapy. He completed his PhD at University of Victoria and was a Canadian Institute of Health Research postdoctoral fellow at University of Alberta. His lab focuses on structural biology and proteins to understand the molecular nature of antibody-antigen interactions with a particular emphasis on nanobodies and antibody-carbohydrate interaction. He's co-authored over 40 publications. Most recently, 3 weeks ago, he just had a new publication, and he holds research grants from the National Cancer Institute of the NIH, CSU Biotech, as well as private research contract from antibody biotech companies. So there we go. Have at it.
Thank you very much, Derek. Everybody hear me okay? I really appreciate the invitation to come down to this beautiful weather. I'm sure it's always like this, right? It's sunny now. I really enjoyed exploring the local parks today for a run. It was absolutely wonderful. So my goal here today is to teach you a little bit about immunology first, and then talk about some pretty exciting new therapies that have emerged really in the past 10 years, which is using antibodies, which are just like our immune system proteins, to treat cancer, as well as other diseases, although my own research really focuses on cancer research. And so I'm going to sort of give you an overview of the immune system, how it works, talk a little bit about the history of antibody immunotherapy, and then, of course, I have to talk about my own work. Professors love droning on about their own super boring minutia. So you'll get to hear all about that. That was all done by graduate students, master's level graduate students, as well as undergraduate researchers. At Fresno State, we have a big emphasis on involving undergraduate research. So without further ado, I'll start in on that. So I like to begin the talk with a couple of disclaimers. So the first disclaimer is I'm a real doctor. I'm not a boring medical doctor. I did a PhD, not an MD. And I like to mention that just because, I'm going to be talking about cancer and, exciting therapies, but I'm really not an oncologist. So I can't give any kind of medical advice. My expertise is really on the, the basic science and not on the telling you how you should get treated for cancer or how your relatives should be treated for cancer. I also like to just start by also disclosing where the money for my research comes from. Some of it comes from a private biotech company, and the rest of it's coming from the federal government, which is the National Cancer Institute. So we're going to begin by giving you a very kind of simplified overview of what happens in an immune response, and then sort of how that goes wrong and results in the formation of a tumor. So your immune system is this amazing machine which is circulating through your body and is constantly looking for problems, basically. And this could be, like bacteria coming in, viruses coming in. It can also be tumor cells forming. And basically what happens is, there we go, there's the laser. If you have something that isn't supposed to be there, the immune system is trained to recognize yourself and then recognize stuff that is not you, right? So that could be bacteria, viruses. It can also be tumor cells. Tumor cells change quite a bit as they undergo the process of becoming cancer cells and effectively become not you, right? So you have this mixture of healthy cells and then abnormal cells. Now, what happens is you also have circulating around your system immune cells. So these your like white blood cells, basically. And what they will do is they will recognize these abnormal cells, right? So maybe a tumor cell, maybe a bacteria, whatever is there. And these local immune cells, they start releasing chemicals into your bloodstream. These things are called chemokines and cytokines. And those are like signals to tell the immune system that there's something wrong and we need to deal with this problem. And so what will happen is the chemokines, they're attractants, kind of like chemical attractants, and they will bring in more immune cells that sort of march in. And this is really like the first line of defense in your immune response. And they're going to come in and try to start killing the things that are not supposed to be there. The cytokine chemicals, they start activating other cells, basically another message saying, we need to start killing things. There's stuff here, right, that needs to be taken care of. Some of the first cells involved, some are called natural killer cells. The name of that cell is really, telling you what it is doing. It is killing things. And so what this will do is it'll kill any tumor cell, bacteria, whatever that happens to be there. And then you also have these things called granulocytes, and they start releasing chemicals that causes inflammation and swelling. And this is why you develop a fever, right? When you start getting sick, it's this inflammation response, which is meant to help kill the stuff that is there. Now, this is where it kind of gets interesting, right? So this initial response of your immune system is really just like dealing with a little localized threat. It's going to kill the one or two cells that happen to be there that are wrong, but it doesn't deal with the whole problem. And the rest of the immune system is this like adaptive response that is going to try to really eradicate the system. So these other cells come in and they basically eat up all of the dead cells. So they will gobble up the tumor cells or the viruses or whatever that was killed by those natural killer cells. And they eat them effectively. They actually engulf them and encapsulate the cell and break it all up into little pieces. And this process is called phagocytosis. Now, what happens is these cells that do this, they're called dendritic cells. They chop up the bad cell, the tumor cell or whatever, and take all the protein and DNA and chop it into little tiny pieces. And they actually like display it on their surface. And then these cells move into the lymph node and show these pieces of foreign material to the cells that are housed in the lymph node. And this includes the B cells and the T cells, which are really the main army that the immune system uses to combat this stuff. All right, so these guys are normally dormant. They're sitting there. They're quite dangerous cells. They're very powerful. When this is deregulated, it causes all kinds of problems. And what these little molecules do is it turns them on, and they start multiplying. They expand. Many, many millions and billions of them will be formed, and then they leave the lymph node and start, going through your circulatory system. The B cells start to produce antibodies, which many people would have heard of, right? This is what happens when you get a vaccine. One of the goals of a vaccine is to produce this antibody response. So these B cells come in, oops, wrong way, there we go. The B cells come in, produce antibodies, And antibodies recognize the bad cells, right? So the cancer cells, the viruses, the tumors, or whatever. And they basically tag those cells to tell the rest of the immune system that we need to get rid of this thing. And those antibodies can then do a bunch of different stuff, depending on the mechanism. They can interact with other cells like T cells, and that will kill the cancer cell. They can bring in more of these cells that eat the cancer cells, and they can actually also activate this toxin mechanism that also kill the cells. There's lots of different ways that the antibody will ultimately, eradicate the tumor or the virus or the bacteria. And again, that's how vaccines work, right? They produce this antibody response. These B cells, they sit in your lymph node and they remember that you've been exposed to this thing before. So the next time it comes in, they can produce a bunch of these. Now, what this has to do with cancer formation is tumor growth is really a failure of the immune system. Cancer cells form all the time. Your cells undergo mutations regularly. You're exposed to UV light and all kinds of things. The oxygen we breathe actually causes mutations. And cancer cells are really just unregulated cells. Most of the cells in our body don't grow. They're highly specialized. They form tissues. And they just kind of sit there for most of your life unless they're damaged and repaired. And really what a cancer cell is a cell that has lost its specialization and it just starts growing uncontrollably when it's not supposed to. Your immune system is pretty good most of the time at taking care of this, right? So a small little tumor might start growing. That's what you're seeing here. You see them all starting to clump together. And all the different immune cells, the T cells and the B cells and so on, are constantly eradicating that system. This fails though sometimes. There's a lot of ingrown systems in the immune system to prevent your immune system from attacking your own cells. It would be really bad, right, if these highly potent killers went around your body destroying your own tissue. So there's a bunch of different mechanisms in place to prevent the immune cells from doing that. And eventually what happens is the tumor cells adapt and they start turning on these signals that tell the immune system, you need to turn off. And so the immune system goes silent and the tumor starts to grow uncontrollably, right? And that is ultimately what results in the formation of a cancer or a tumor. All right. So what is antibody immunotherapy then? This is basically the idea of, well, if our immune system is failing in combating the tumor, is there a way we can either reactivate the immune system or sort of give it some help to, turn it back on again so we can remove the tumor? Now, this sounds like pretty advanced technology, and it is, but it's actually a very old idea that has really only recently come to fruition. You can go back all the way to the beginning of the, all the way back to the beginning of the 20th century. And the original idea of this comes from these two gentlemen here, Paul Ehrlich and Emil von Behring, two German physicians. And they came up with this idea, what they called the magic bullet hypothesis. This was Paul Ehrlich's idea. And he envisioned some sort of molecule that you could have that would act like a bullet out of a gun that you could use to, kill a bacteria or a virus. In the early part of the 20th century, this is really what they were concerned about, right? We had polio and smallpox and all these horrible things. Vaccines didn't exist yet. And so they were really interested in, especially infectious diseases. And what they observed was if you took diphtheria, which is a bacterial toxin, and put that in an animal and actually extracted the blood from that animal, it would effectively neutralize the bacteria. And so Paul Ehrlich thought that this was his magic bullet, right? This is the cure I've been looking for to get rid of diphtheria. And they sort of hypothesized that there were cells inside of this serum that had something they called side chains. They didn't really know what it was. And those side chains were the things that were, killing the cells. And they didn't know it at the time, but they were actually describing antibodies, right? So these guys had described and, effectively discovered antibodies, 50 years before it was actually, really discovered. And so this was used quite heavily in the early part of the 20th century. You would basically immunize, take diphtheria toxin, put it in an animal, harvest serum, put it in a bottle, and then that would be used as a treatment if you contracted diphtheria. And so this was the very first immunotherapy. There were some problems with this, though. The biggest problem was, what they're doing is using horses. Horses are large animals, and injecting this stuff into a horse. And not every batch was the same, right? So sometimes the serum worked really well, other times it did not. And you may remember that in the COVID outbreak early on, when there were no treatments, one of the very first things that was given to patients that were dying was convalescent serum, which was basically serum taken from COVID patients who had survived, and they gave it to other people. So basically resurrecting this like really old idea. This was basically abandoned, though, in around like 1923, 1920s, when antibiotics were discovered, right? So these molecules were so effective at eradicating bacteria that they basically didn't need this treatment anymore. And the idea was sort of forgotten for a really long time. And then in the late 70s, early 1980s, a major breakthrough came, which was the invention or discovery, I guess, of these things called monoclonal antibodies, which the 1984 Nobel Prize was given. And this is an absolutely ingenious idea. And the way that this works is you want to generate an antibody against a particular thing, you immunize a mouse. So you take your tumor cell or your bacteria or your virus or whatever, and you inject it into a mouse. And then that mouse is going to have a normal immune response. It's going to start making antibodies. And in the old days, they would just harvest the serum. But to get over this problem of every time you do this, it's different. What they did is they took the mouse and they extract the spleen cells. And inside of the spleen cells are the cells that are actually making these antibody proteins. And they took myeloma cells, which are a type of tumor cell. It's a blood cancer, myeloma cell. And you mix the cells from the spleen with the myeloma cells together, and the cells will actually fuse together. So you now have a cell with like multiple nuclei that is a hybrid cell between this blood cancer and the spleen cell. And what that does is it makes the cell immortal. So it will live forever and continue to grow and grow and grow. And it's very easy to grow it in the lab. So once they made these hybrid cells, which they call hybridomas, they then can grow them forever. Those cells are secreting antibody, and now they can purify an infinite supply of pure antibody from this mixture. And so it was very quickly realized that this could be a way of generating new drugs, right? So you immunize the mouse against whatever disease you want to target, make a hybridoma cell, make the antibody, and then administer the antibody as a drug. And this has been done successfully. The very first one was approved by the FDA in 1997. And this is a sample of many of the types of drugs that you would see out there that are monoclonal antibody drugs. Drugs typically have two names to them. They have kind of like a common name. So up here we have Humira, which is sort of like a brand name. And then they have the chemical name. So this one is called Adalimumab. And so whenever you see that MAB at the end of the drug name, that is a monoclonal antibody drug that was originally isolated using this like hybridoma type technology. Now, it turns out the way that we can use these antibodies as drugs is pretty variable. There's actually a lot of different mechanisms by which this can work. The simplest and kind of most obvious mechanism is the antibody can work by activating the immune system. Right? So your normal immune system has failed during the development of the tumor. So what you do is you generate an antibody against a particular tumor and kind of re-inject that back into a person. And then that reactivates all of those immune cells that were turned off. Right? And then those immune cells now can go in and start to eliminate the tumor. So that's the one way that this can work. An even more clever way that this can work is you can use this instead of a way of changing the immune system. You can use it as a delivery system, sort of like a nuclear missile, right? You like use it as the missile to deliver the nuclear bomb to the tumor cell. So what they do is they generate an antibody against a type of tumor and then attach to that tumor like a radioisotope, right? So a radioactive compound that is emitting beta radiation. And so when the antibody comes down onto the tumor cell, it is irradiating the tumor cell only at the site of the tumor, killing the tumor. Or you can attach a normal chemotherapy drug to it. And what it's going to do is it's going to bring the chemotherapy drug directly to the tumor cell instead of going to all of the cells in the body, again, killing the tumor. A third way that this can work is by stopping signals. And so when the cancer cell is growing, it is adapting to its new environment, and it is turning on all these messages to tell the cell to grow, which is the thing that is causing the tumor to exist in the first place. You can silence those messages by targeting the receptors that cause that to happen. That's a third way that these drugs can. And so since that initial discovery or approval in 1997, there's been basically like an explosion in the use of these drugs. And so this is looking at a graph by year of FDA and European Union approval of antibody drugs. And you can see it's kind of like a trickle at first, right? So 1997 is the first one, and then there's kind of some in the early 2000s. And going into our current decade, the line is going up quite exponentially. And about half of these are used for cancer therapy. And this is a breakdown of the current number of FDA-approved monoclonal antibody drugs. There's currently 206 on the market. About half of them are for cancer. The other big target is immune disorders, and then kind of a bunch of other different stuff. So in terms of how effective this is an empirical question and an interesting question. And so I wanted to just show you kind of like where we stand in terms of dealing with cancer, kind of state of the art of it. In terms of cancer rates, how many people are getting cancer, the rate of cancer incidence is actually pretty flat. There's a misconception that the amount of cancer is increasing in the modern time. That is not actually correct. It is more or less flat. Some cancers are going up, some cancers are going down. The net effect is it is roughly flat. One positive, though, is the death rates are going down. So again, this is just looking at kind of all the different types of cancers and the 5-year survival rates. There's been really big progress in certain ones. Lung cancer, probably the reason why lung cancer is going down so much is people don't smoke as much as they used to. Melanoma is another one that has gone down enormously. There's some that have gone up, but the net is it is decreasing, although small. So you could say that maybe treatments are helping. These new treatments we're developing are bringing the death rate down. And early diagnosis is obviously very, very important as well as lifestyle. Now, the traditional way that tumors and cancer are treated is this, what they call the 3 pillars of oncology. The first is surgery. And so if the tumor can be just cut out, that is highly effective a lot of the time, right? And certain cancers can be easily treated by surgery. The second pillar of this is radiation, right? So you simply irradiate the tumor with beta radiation, which kills everything. And then the third is chemotherapy. And the way that most chemotherapy drugs work is they're going after DNA replication. The cancer cells are rapidly multiplying. Most of the cells in your body are not rapidly multiplying. And so these chemotherapy drugs inhibit this making of new DNA. And of course, there are some cells that are always growing, like your hair follicles. And so the chemotherapy drugs kill the hair follicles, right? And that's one of the reasons why hair falls out during chemotherapy. So where does the antibody or immunotherapy kind of fit into this, bigger picture of these 3 types of things? So I wanted to give you one example where this has had very large and dramatic effects, which is a type of disease called metastatic melanoma. So melanoma, of course, is a type of skin cancer. Metastasis is when the tumor spreads. So what often happens is a primary tumor forms at some tissue, so in this case, a skin cell, and they will, as they grow over time, they will eventually detach and spread through your circulatory system. The tumor cells get deposited in different places and start to grow in other places. When this happens, as I'm sure many of you know, this is extremely serious. That means that surgery won't work anymore. You can surgically remove the spot where the skin cancer is, that's fine, right? It's gone. But if it is spread through your circulatory system, it's just going to grow back again. And metastatic melanoma prior to 2014 had a survival rate of 15%. So that is an extremely low survival rate. It is almost a death sentence, right, if you're diagnosed with metastatic melanoma. And then around 2014, 2 antibody drugs were approved by the FDA. One is called nivolumab, the other is called ipilimumab. And this was a very clever application of these drugs. And what they do is they basically turn off that signal that tells the immune system to go to sleep, and it like hyper reactivates the immune system. What's going on at the tumor interface is the tumor cell is actually showing stuff to your immune system, and it is telling your immune system to turn off. There's a protein that it sits out in the surface, and it says, T cell, you're not supposed to kill me. I'm supposed to be here. And that silences the immune system. This nivolumab antibody, all it does is it blocks one of those receptors that reactivates the T cell, and now the T cell can start to kill the tumor. The other antibody drugs work a similar way, but just against a different type of cell. And so double treatment with these 2 things basically completely reactivates the immune system, and it will start to clear the melanoma. And just to give you a sense of how well this works, the 5-year survival rate was 15 to 20%. Double administration of that drug brings the survival rate up to 52%, right? So it's a massive improvement. What used to be a death sentence is now like a flip of the coin, which doesn't sound that great, but it's a lot better than a absolute death sentence. Now, for reasons that are not entirely clear, this does not work with everybody, right? For some unknown reason, there's a fraction of the population that is effectively unresponsive to this treatment. The Nobel Prize was given for this idea, I think a few years ago in 2018 for discovering this system of reactivating the immune system. Now, one, I would say, big downside to antibody drugs is how expensive they are. The market for antibodies is extremely profitable for pharma. This is just showing you the amount of dollars, net dollars in the United States. Oh, this is global, sorry, for the antibody drug market. So that's in billions of dollars. So last year it was $250 billion was the market size of that drug. The average cost for 1 year treatment is $150,000 for the antibody drug, the extreme cost. The reasons for this, which the drug industry give, has to do with the extreme expense of developing these things. The average cost of bringing a new drug, antibody or otherwise, to market is $2.5 billion, right? So it is a massive investment for these companies to do this. And the reason why they charge such enormous price is there's a huge risk for these companies to invest this much money in a drug which may fail, right? It may not actually go to market. The reason why it costs so much is because of the very long time scale in going from the initial discovery of the drug to the final approval by the FDA. And so this gives you like a sense of the time scale here. So from the beginning of the project, when the company sort of decides we're going to build an antibody that targets this cancer, it takes 1 to 3 years to sort of go through the discovery process, find the antibody, do some initial testing, show that it works on cells, do some like initial safety tests. This also then goes into animals, would be the next step. So these are injected into mice and guinea pigs and things like that, to find out whether it can cure cancer in the guinea pig first. And then the really expensive part is when this goes into clinical trials, which is the administration of the drug to people, right, on a trial basis. For a drug to be approved, it has to go through 3 levels of testing. Phase 1 just tests whether the drug is safe. So it is first given to healthy people, and then they just record how many people get side effects, how bad are those side effects, and so on. If that looks okay, it goes into phase 2, which is a small number of people, kind of like 50-ish people, that type of scale. And you have a mixture of healthy people, sick people. Some people are given the drug, some people are given the placebo, and then you figure out whether the drug actually works. And then the final stage, phase 3, is the very expensive one. It requires often tens of thousands or thousands of people to be administered the drug with placebo and without, and costs millions and millions of dollars to do this. And so this is the main reason why this takes so long and is so expensive. Which again, in the COVID times, was something that was sort of passed over, is how quickly some of these drugs were made and approved, less than 2 years. And so there can be kind of like a lag between the basic science and the drug development. And so this is where I'll bring it to my own boring little niche of research and tell you a little bit about what basic science we can do in the lab without billions of dollars. For context, my research grants are more like $300,000, not $2.5 billion. And really what small research does is it lets us sort of like think a little bit about what might be a good idea, right? Do some initial testing. Maybe we get some new information from these studies. We can decide, is this still a good idea to do this, activity? I don't know. Maybe not. So my lab is working on several projects related to pancreatic cancer. Pancreas, right, is a small organ, part of your digestive system. It's involved in producing some hormones, also in like digesting protein. This kind of like funny looking little organ. It's not a large number of people that are diagnosed with pancreatic cancer every year, small number, less than 10,000 per year in the United States. But it has a extremely low survival rate, less than 12%, right? So again, especially if you're diagnosed with stage 4 pancreatic cancer, it's quite severe, right? The reason why the survival rate is so low is the symptoms don't really appear until quite late, right? So you could have pancreatic cancer, you basically feel more or less fine, right? You might feel a bit off, you don't go to the doctor, you know, who wants to go to the doctor? And then by the time you have severe symptoms, it is at like stage 4, where it is spreading through your body. The main treatment currently is just surgery. The most effective treatment is surgery. So if it's caught early enough, the pancreas is just removed. And so the idea that my lab has been working with, along with a couple others, is we want to make an antibody against something that is sort of unique and specific to pancreatic. The cells that are part of the pancreas have this layer on them made of these proteins called mucins. And the reason why that word sounds like mucus is because that is what mucus is. Mucus is actually a protein that is like covered in sugar, and it like absorbs a bunch of water and makes it that kind of gross, like mucusy surface. And the surface of the pancreas is basically lined with these proteins called mucin, which are covered in sugar, right? So that's what the little squiggly lines are, the protein, and then the little blue squiggly lines are sugars that are like coating the entire thing. And it basically acts as just like a lubricating layer on the surface of the cell. Now, what happens in the formation of pancreatic cancer is there's a bunch of big changes that happen to those mucin proteins. They start to lose a bunch of their sugars, and they start to be secreted and sent out into the serum. And so that means the protein is different on the cancer cell versus the normal cell. And if it's different, that means it's something that we can target specifically. So this protein called MUC16, this absolutely massive protein, the second largest protein in the human protein system. And it's just made of these repeating units over and over again that are covered in sugar. Now, the reason why this protein is a very useful thing to target for treating pancreatic cancer is recent research has sort of shown that this seems to play a really like central role in regulating how the tumor grows. The protein kind of sits here in the membrane, and it interacts with all these other proteins, right? So these are other proteins here that are all binding to it. They all like stick together, and that turns on a whole bunch of signals that activate a whole bunch of genes, and the cell starts to grow. So it's sort of the main signal that lets the pancreatic cancer cell grow. And it's involved in all kinds of stuff. It's involved in the separation of the pancreatic cell, so it starts circulating the growing of the cell. It's quite important. It also seems to turn off the immune system. The MUC16 protein, again, interacts with some other proteins and starts to suppress the immune system. So it seems to be a super important molecule kind of regulating the cancer cell. So we've been working on a monoclonal antibody. It's called AR9.6. I have no idea where that name comes from. And the antibody is held by this small little biotech company called Quest. It has 3 employees, very small little company based in Edmonton, and they own the intellectual property to this antibody. Now, the story behind this like all good science stories, was a accidental discovery. I have a friend and colleague at the University of Nebraska Medical Center in Omaha. He was working on this project, completely unrelated to trying to discover a cancer drug, and sort of on a offhand, random experiment he just sort of decided to do. He treated some pancreatic cancer cells with a bunch of different antibodies. And this one antibody in that panel, AR9.6, seemed to turn off the cell signaling that controls the tumor cell growth. He then went on, he thought that, well, that was very surprising. Maybe we could use this as a pancreatic cancer drug. So he went off, and you can actually take a tumor cell, implant it onto a mouse. It will start to grow a tumor on the mouse. So he did that, and then treated the mice with the antibody drug, and it caused the tumor to shrink. Right, so that's what this little graph is here, is he combined it also with a chemotherapy drug, and all of a sudden the cells stopped growing, implanted the mice, and then the tumor starts to shrink with these little dots over here. And this was basically the stage where my lab got involved. The original antibody was made in a mouse, which is the original technology. So someone many years ago injected a mouse with some ovarian cancer patient tumor cells and generated this antibody. The problem, though, is the antibody comes from a mouse. And so if you inject that into a person, your immune system actually thinks that this is not supposed to be there. It's a mouse protein. Why is there a mouse protein here? And attacks it and removes it. And so one thing that has been done is we can take a mouse protein and basically turn it into a human protein through this process called humanization, which is like nicely illustrated here with this cartoon my graduate student developed, where you have a mouse, and then first you make it into a chimera, right, which is sort of like a half mouse, and then you humanize, which is an almost, human, still some mouse features, and then hopefully you can make it fully human. And so the way we do this, kind of in brief, is we take those cells that are producing the antibody, the original mouse cells, we sequence them, figure out what the sequence of the antibody is, and then we go into a database and start to look at human proteins and start to make changes to make it more and more like a human protein. We then synthesize the DNA and stick it into some other cells, and then those cells start to make the human-like protein. And then sure enough, we're able to make the protein right there. We're able to make tons of it. And then our protein, our human one, basically behaves the same as the mouse one. That's good. That's very similar to the mouse protein. The other thing that my lab does is we look at the three-dimensional structure of proteins. We use this technique called X-ray crystallography, which allows us to actually look at every single atom inside of the protein. So you get these highly accurate three-dimensional models of what the protein actually looks like. And that's what you're seeing on this slide. I know this is like a fancy cartoon, but this is some graduate students' work. And from this, we found that, again, like our protein, the humanized one, looked almost identical to the mouse one. So that was great. We then started to figure out where this thing binds on the protein. So we took the original MUC16 protein and broke it up all into pieces, and we're able to figure out roughly where it bound on the structure of the protein. And then we were lucky enough to get another structure of the antibody with the actual mucin protein again. So now we know exactly where on the structure it binds. All right, so that's all fine. It's all very interesting, very academic. The main takeaway from this, though, is we know where the sugars are supposed to be on that protein. So we build the model, looks like this. And it turns out the antibody actually binds very close to where some of these sugars are. And this actually gave us a very important clue as to how this antibody actually works. So it turns out that this protein sitting here, this MUC16 protein, binding all these other little components over here, it uses sugars to do that. So it's displaying these small little sugars. Those little sugars get bound by these other things, and that turns on all these pathways. So what we think is happening is the antibody is interfering with the sugar binding. So that is going to block the interaction between those. And then that in turn is going to block the signaling. That causes the tumor to start to shrink, right? Because it's losing the signal to grow. The other things we've done with this, a little bit more visual, is we work with a couple other labs. We've taken our human antibody and worked with these other labs to do some other rather interesting applications. So Aaron Mose, University of Nebraska also, he's interested in using this to help surgeons more effectively remove the tumor cell. So what he has done, he takes our antibody and he conjugates other molecules to it, which glow under UV light. They fluoresce. So they produce this like very bright glowing signal. And so what you're looking at here, again, is a mouse that's been implanted with a tumor and then has been treated with this antibody. You can see that glowing dot. That is the tumor glowing under the fluorescent light. So the idea here is, now if you're a surgeon, you need to remove every single piece of that tumor, because if you leave a piece behind, it is just going to grow again. And so the idea is now you can put this under an imager while you're doing surgery and completely remove the tumor. And so this is a little, and you can see it's like glowing right there, a big tumor. I have a short little video of the graduate student doing some surgery on a tumor that's glowing under light. Don't worry, it's not that bright. You see that glowing light there? That is the actual pancreas tumor being surgically removed under this like bright light. And so you can see how effective that would be, if it went to market. Kind of a similar idea, different group at the Sloan Kettering Cancer Center. They're more interested in diagnosis and imaging applications. So they've taken our antibody and linked a radioactive molecule to it. And now what you can do is put in this case, an animal with a tumor, into a PET imager, a positron emission topography, which is one of the ways that we diagnose cancer. And it will basically just light up the tumor, right? You can see the tumor glowing brightly under this imager. They're also in the process of attaching a second radioactive molecule to it, with the idea being the second radioactive molecule will actually irradiate the tumor and hopefully kill it. And so in terms of monoclonal antibodies, like where do we actually, stand in the whole field? I would say in some cases, this has had like spectacular results, especially with the checkpoint inhibitor, metastatic melanoma. However, most of the time, what you are seeing is really this is just enhancing outcome by a small fraction. It's just an additional thing that is going to be done in combination with chemotherapy, radiation, and surgery, and maybe improves outcomes by 5%, 10%, something like that. But those 5% mean a lot, right? If it's someone you love that is being treated with these drugs. So they're not really a miracle cure, but they're an extremely useful, tool in the toolbox that we have to fight cancer. And I would say there is very big excitement in the air on these. There's lots of new ideas and innovations that are being done. People are combining different antibodies together to make hybrids and inventing all kinds of weird formats. And so there's big investments being put into this, partially because of the large profits that companies make from the drug. And so I expect that we're going to see many new drugs coming out in the near future. And so this work, of course, I don't actually do most of this work. Professors tend to just sit in their offices writing grants and writing papers. It's actually mostly done by my students. So my lab, a handful of graduate students did most of this work, as well as with collaborators at the University of Nebraska. I didn't talk about these guys, but the National Research Council, they do a bunch of stuff for us. And the CEO of Quest has been very supportive of us doing this research on this antibody drug. And so with that, I'll take any questions you might have, and I appreciate your attention.
All right. So if you have any questions, please raise your hand and I'll come by with the microphone. That way people watching remotely will be able to hear it as well.
No one's brave.
All right, here we go. That's all wins first.
I'll preface this with saying I only work on dead animals. I'm a paleontologist.
Okay, yeah.
But so besides just that, like everyone works on mice, would there be like a better thing closer for developing like why don't we use primates? Is that just ethics?
Yeah, so there's in the cancer field, there's a lot of discussion about the models that are being used. A lot of early work, especially like 1990s and the early 2000s, was done heavily on mice. And what they would do is they take a human tumor, implant that onto the mouse, and then do the study. And there was big problems with that because, as you say, it's a mouse, not a human. And so it has a mouse immune system, which is not the same as a human immune system. And in many, many cases, there was lots of excitement where people were like, we cured cancer. And then it goes into human trials and it doesn't work. And since that time, there's been big investments in basically making mice a lot more like humans. So they have generated transgenic mice, which now have a human immune system instead of a mouse immune system. They no longer use tumor cells, which they have been growing in the lab forever. Now, what is often done is the tumors are actually taken from patients that have recently died in autopsies, and they take those tumors, so real human tumors, and those are implanted. And those studies tend to translate like much better. And people do monkeys, but monkeys are expensive and hard to work with.
I have two questions. First one is, how does one generate a synthetic antibody or part of a protein generate? Do you actually design it and sequence it from scratch, or is it that a cell is generating lots of them and you select the one you want?
Yeah, we're selecting. So when you generate these hybridomas, there's millions of cells, and each one is like secreting a unique antibody. And a lot of the work is isolating individual cells that are producing an individual antibody. And then we sort of screen that, try and find the one that, or the handful of ones that look the best, and then we sequence it. And then from there, then you can move it out of the system into a different system.
And second question, I forgot the name, but that very large protein that the AR,
Yeah, MUC16.
Many years ago, I was told that it's difficult to crystallize out of large floppy proteins, especially ones that are in membranes, especially ones that interact with other molecules. So I'm wondering, how do you do it? It seems really hard.
So that one, it's nice. It's like a repeating unit. So it's
You did
a small unit that's repeating. So we just take one unit.
Oh,
And we remove some of the extra stuff, the sugars and things. And so that's much easier. Piece by piece. The whole thing would be probably impossible.
See if my voice lets me pull this one off, but in terms of the cancer immunotherapy, are there cancers that are more likely to respond to this sort of treatment, or are there just certain ones that have been tried?
Yeah, that's a great question. In general, this works best with like blood cancers, cancers that are not very solid. The really solid tumors, this does not work very well. The solid tumors get really, really large and they're very dense, and the antibodies can't penetrate very far into the tumor. So there's been not a lot of success with this. So this works really well with some and very poorly with others.
Oh, hey there. You mentioned that your research was mostly into an antibody therapy. Is there any research being done into a modified T cell therapy?
Yes, that is a whole other thing, which is also very exciting. There's a bunch of different things there. There's these things called TCRs, and basically what you do is, like I said, in what happens in the normal immune system is your T cells get turned off. And so what they do is they actually harvest T cells from the patient and sort of sort through those T cells and find the handful of ones that are active, regrow them, and then inject them back into the person. And this is extremely effective, but insanely expensive, right? Because of the work that is required. It's very specialized. It, works for you and just you, and requires a huge amount of work. So it's like $300,000 for one treatment.
Yeah, there was that study out of Israel about a year ago. There was that I think 85% total recovery rate, but the sample size was pretty small. I was wondering, you mentioned antibody therapy was really expensive, but I'd imagine this is much more expensive. Okay.
And there's also some really bad side effects sometimes with that. There's this thing called a cytokine storm.
Okay, we have a question from online. Angam Ahmed had asked, hello, if a person raised an antibody that is specific and has high affinity to a tumor, can you produce this antibody to treat a patient with a tumor instead of starting from scratch?
Sorry, that was quite long. Can you repeat that?
Absolutely. Okay, I'm just going to read out how it was.
Yeah.
If a patient raised an antibody that is specific and has high affinity to a tumor, can you produce this antibody to treat a patient with a tumor instead of starting from scratch?
Yes, you could do that. That's the short answer. Angam is my former graduate student. She's watching.
Any other questions? All right, I guess that will complete our first chemistry lecture series. Thank you very much.